Hello and welcome to the first video of a few-part lecture series about Chapter 16, Molecular Basis of Inheritance, or AP Unit 6, Gene Expression and Regulation. In this video, we're going to talk about the first two subunits. First off, DNA and RNA structure, and we're going to dip our toes into replication. Okay, so it might sound obvious to you now, but for a long time it wasn't understood that DNA was the source of heritable information. Which means that genetic information is stored in the sequence of nucleotides that make up DNA. We can also, briefly summarize this by calling it the DNA structure. And these specific nucleotide sequences are is what's passed through generations, from parent to offspring, and this can only happen by replicating that parental DNA. So we can summarize this by calling it DNA replication. All living things, from the tiniest to the biggest, the least complex to the most complex, all pass their heritable information through these same 4 or 5 nucleotides, which is just crazy. But let's first compare the genomes of prokaryotes and eukaryotes. So in prokaryotes, their genetic material, which can be DNA or something called RNA, is circular. So their genomes are typically of a smaller size. They have less base pairs than eukaryotes. And they have plasmids that in prokaryotes are located in the cytosol. What's a plasmid? Here you can see it's a small double-stranded DNA molecule. It's circular that exists outside of the chromosomes. So here's the bacterial double-stranded DNA, but also contained within the cytosol is the prokaryotes plasmids. So just think of this as like an extra little bit of DNA. Now looking to eukaryotic genomes, their genetic material is made up of multiple linear chromosomes. Eukaryotic genomes are typically of a larger size, and some eukaryotes do contain plasmids, yeast for example, but their plasmids are contained within the nucleus because they store their genetic information within a nucleus because they are eukaryotes. Okay, like I mentioned earlier, we were basically clueless as to the 3D structure of DNA. I say clueless, like they had a few clues, so we understood that DNA was a polymer of nucleotides, meaning just like a long string of nucleotides, and we knew what these nucleotides, what the structure of these nucleotides were. So here it is right here on your right, just kind of a generic structure of a nucleotide. So you have a sugar, a phosphate group, and a nitrogenous base. Now, they also realized that in DNA, there's only 4 possibilities for nitrogenous bases, which are your purines and your pyrimidines. So in the purine category, we have adenine and guanine, and then in the pyrimidine category, we have thymine and cytosine. We got a little further in understanding what the possibilities of the 3D structure of DNA could be with Chargaff's rules. So in 1950, Irwin himself realized that DNA composition varies between species. So we have different percentages of these nucleotides in like grasshoppers and octopuses, for example. But of course, there is these same levels of differences in between all species. There's molecular diversity among all species. He also realized that there is some regularity in these ratios, no matter what species you're looking at, or what, animals, plants, and bacteria alike, these same ratios appeared. So he noticed, for example, that roughly about the same amounts of adenine and thymine existed within every genome, and roughly the same amounts of guanine and cytosine existed between all genomes. So 2 things really became known as Chargaff's rules. So one, that the composition of DNA, the base composition of the nucleotide sequences varies between species, but he also realized that no matter the species, roughly the number of adenine and thymine is equal, and the number of guanine and cytosine is equal. While these 2 findings became well established as Chargaff's rules, the basis for these rules was not really understood until later. So we also had another clue, which was how these covalent bonds were arranged within either DNA or RNA. So these covalent bonds exist between the phosphate group of one nucleotide and the sugar of the next nucleotide. So here you can see the phosphate group, which is denoted with this yellow color, is connected to this pentose sugar of the next nitrogenous, or of the next nucleotide. And because we understood how these nucleotides fit together, we also could tell what was at the ends of them. So there was a phosphate group on what we call the 5' end. You can see here that they number the different carbons of the sugar, so we can kind of like label where everything is. So we knew that there was a phosphate group at the 5' end of the nucleic acid, and that there was a hydroxyl group, which the chemical is written with an OH, at the 3' end of the nucleic acid. So we knew how the nucleotides were bonded and fit together, but we still didn't understand the three-dimensional structure of DNA. Of course, there was lots of people that were trying to figure this out. One of these people was Linus Pauling. So Linus Pauling thought that there was this structure of DNA where the nitrogenous bases were on the outside and the sugar phosphate backbone was on the inside of the structure. Of course, he didn't end up getting famous for being right about that. Instead, he was famous for being like a big vitamin C advocate. I don't know. The 4 people that really ended up figuring this out were James Watson and Francis Crick, who kind of worked together, and Maurice Wilkins and Rosalind Franklin, who also kind of worked together. So it all really started with Rosalind Franklin's famous Photo 51. Photo 51 is not actually a photo, but it came from Rosalind Franklin's really premier skill of X-ray crystallography. So how this works is she would shoot X-rays down at the structure of DNA, and these smudges and smears that you see in this photo could help you understand what the chemical structure was of whatever you were looking at, which in this case was DNA. So that was Rosalind Franklin's contribution. She took this X-ray crystallography photograph, and the person who they shared a lab with, Wilkins, and her were trying to figure this out, but couldn't quite figure it out. The 2 that came along and finally literally put the pieces together were Watson and Crick. So from Franklin's X-ray diffraction photograph, Watson and Crick could deduce this double helical model and understand how the nitrogenous bases actually fit together in the way that we are so used to seeing today. So there were a few specific findings of Watson and Crick's that came directly from Photo 51. One of these is they realized that DNA had to exist in a double helical structure. They could also understand the width and the spacing between not only the individual base pairings, but also the turns of the 2 strands. They also understood that the 2 strands run in opposite directions, meaning they are anti-parallel. So here is a simplified version of the 2 strands of DNA. You can see that the 5' end, which is the phosphate group, remember, and the 3' end, which is the hydroxyl group, lay next to each other. And they also got closer to understanding how the base pairing rules really work, because at this point we still don't understand how Chargaff's rules operate in real life. So at first, Watson and Crick thought that the bases pair like with like, so adenine with adenine, and so on from there. But other pairings didn't really result, but these pairings didn't really result in a width that would make sense based on the crystallography photo. But then also it was just like clunky. They were just too big or too small in certain places. So instead, they figured that the adenine and the guanine and the cytosine and the thymine, or only purines with pyrimidines together, will result in the uniform width that's consistent with the data from Photo 51 that we see here. So again, purine and purines were too wide, the pyrimidines were too small, but together it finally made sense. And now Watson and Crick's model really explains Chargaff's rules. So in any organism, in any species, the amount of adenine roughly equals the amount of thymine, and the amount of guanine roughly equals the amount of cytosine. So here again, we see our purines, adenine and guanine, bonded with our pyrimidines, thymine and cytosine. Also, I want you to notice that the A and T pairing has 2 bonds right here and right here, and the guanine and cytosine pairing have 3 bonds. So all of Watson and Crick's findings, the double helix, the width and spacing of the bases, and the final understanding of Chargaff's rules was all detailed in a single-page paper released in 1953 in the journal Nature. It's pretty crazy that it's only 1 page. You know, you would think it'd be like so complicated. 1 page. Now, before we go any further, I want us to back up to the first slide and remember that we said the source of heritable information is DNA. But I want to point out here that sometimes heritable information is stored in RNA. So RNA is just another way for organisms to store their heritable information or their sequence of nucleotides. There's a few differences though between DNA and RNA. One of them being that RNA is single-stranded, and another difference being that it uses uracil in the place of thymine. So uracil and adenine will bind in RNA instead of the adenine and thymine that usually binds in DNA. So it was in the last few sentences of Watson and Crick's paper that they disclosed the following information. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So what they're saying here is that we know DNA has to replicate itself to make it to the next generation. So the basic principle of DNA replication is that the parent strand kind of acts as the template for the 2 new strands that are created. So how this occurs is that the parent strand, or this original double helix, will unwind and split, and then 2 daughter strands will actually be built based off of that, based off of one of the parental strands. So here we see parental DNA. It's double-stranded. It is unwound, but it hasn't started actually replicating yet. The 2 strands will separate, and then those 2 strands will each become templates for the newly forming strands. So because we have the complement bases, according to our base pairing rules, we can form these next 2 brand new strands. So what this really all says is that DNA replication is semi-conservative, meaning that when a double helix replicates, each new daughter strand will have 1 piece of the original parent strand. So it's semi-conserved. There was a couple other competing models, like the conservative model. So somehow the 2 parental strands will come back together after replication. Of course, we know that that is not the case. There was also something called the dispersive model, in which like pieces of the parental molecule will be dispersed throughout the newly formed molecules, which again ended up not being the case. So we realized through a series of experiments that DNA replication is indeed semi-conservative. So DNA replication is incredibly fast and incredibly accurate. DNA can replicate itself at a speed of about 50 base pairs per second, and this replication has to happen every single time a cell divides, and cells divide around 2 trillion times a day. So there's a lot of DNA replication happening, and every time most of the time it is incredibly accurate. The molecular machines that make DNA replication possible include dozens of enzymes and other proteins, but luckily, we are only going to focus on 4 of these, at least for now. So helicase is an enzyme that actually unwinds the strands. It's in charge of unwinding the helix, and then topoisomerase will relax the supercoil. So it gets like really tight right around where it is starting to uncoil. So topoisomerase kind of like relaxes that supercoil at the replication fork. What's the replication fork? That's like this Y-shaped formation where the strands are actually separating, like where the replication is happening. DNA polymerase is another enzyme. What its job is to synthesize new strands. So this is the enzyme that actually builds new strands, or like builds our newly formed strands based off of the template. We'll talk more about what RNA primers are, but they require RNA primers to attach to certain parts of the DNA molecule and start the synthesis process. The only place where this DNA polymerase enzyme can attach to the strands is at the 3' end. So from that, we can understand that DNA replication builds in a 5' to a 3' direction. So it's going towards the 3' end. And then one more enzyme that I'll mention is ligase. So ligase joins fragments on the lagging strand, which we'll get more into later what lagging and leading really mean. So what's the crucial information from this lecture video? One is that DNA and sometimes RNA contains genetic information, and that information can be passed through generations. Prokaryotes have circular DNA or RNA, and eukaryotes have multiple linear chromosomes. Plasmids, which are double-stranded circular, like extra bits of DNA, exist in both prokaryotes and eukaryotes. There are specific base pairing rules between the purines and the pyrimidines. Remember that purines are a double ring structure and pyrimidines are a single ring structure. The other thing that we should know is that DNA is synthesized in a 5' to 3' direction. And this replication is semi-conservative, and there are several enzymes that carry out this replication, including helicase, topoisomerase, DNA polymerase, and ligase. All right, fireworks! Next time, we will talk about how these enzymes really work together to replicate each DNA strand and just go a little bit deeper into DNA replication. Thanks. See you next time.